Manoj, you are muted.
Yes, sir.
Dr. Bridges has joined.
Okay. So, with this permission, shall we start, sir?
Yes. Bridges, sir?
Yeah, he has joined and
Manoj, shall we start? You are muted. Dr. Bridges has joined.
yeah.
Welcome to MC Music World. The Bluetooth device is ready to pair.
Bridges, sir?
Connected.
He has joined and
Manoj,
okay.
shall we start?
Yeah, sir. Okay. So, good afternoon.
Welcome to MC Music World. The Bluetooth device is ready to pair.
Bridges, sir?
Connected.
Shall we start?
Yeah. Okay. So, good afternoon. Good afternoon, sir. So,
माझा येते का आवाज?
Yes, yes. Hello? Bridges, sir? Bridges, sir? Bridges, sir, you are not audible.
माझा येते का आवाज?
Yes, yes.
yes.
Hello?
You are audible, sir.
Bridges, sir? Bridges, sir? Bridges, sir, you are... मनोज, आवाज नाही येत आहे ना?
नाही, तुमचा येतोय.
ब्रिजेश सरांचा नाही येत ना?
हॅलो.
मनोज, आवाज नाही येत आहे ना?
येतोय, सर. येतोय.
ब्रिजेश सरांचा नाही येत ना?
सर, त्यांचा पण येतोय. येतोय. सर, त्यांचा पण येतोय.
हे रिपीट का बरं होत आहे सगळे?
Yeah. So, good afternoon, all. On behalf of SNJB Sriman Suresh Dada Jain College of Pharmacy, Chandwad, it gives me immense pleasure to warmly welcome all the delegates, distinguished resource person, faculty members, research scholars, and dear students to day 5 of this and the concluding day of this 5-day online faculty and student development program entitled Innovative Approaches and Emerging Trends in Pharmacology, Teaching, Research, and Practice. Over the past 4 days, we have witnessed enriching deliberations, insightful technical sessions, and meaningful interactions covering diverse dimensions of pharmacology, from pedagogy and research methodology to experimental, molecular, and industry oriented perspectives. The participation and engagement from all of you have truly made this FDP SDP a success. On this occasion, okay, the 90th birth anniversary of late Bausa Sampatlalji Pemraji Surana, we pay a great tribute to this great personality and proceed further with our today's session, which is based on the theme professional development and translational pharmacology from bench to bedside. The focus of this technical session is on the area that is crucial for academicians, researchers, and students alike: grant writing and funding opportunities. Securing research funding is an essential skill for translating innovative ideas into impactful scientific outcomes. The technical lecture for today is on grant writing and funding opportunities for pharmacology research based on ICMR, DBT, and DST. This session aims to provide practical guidance on preparing competitive research proposals and understanding national funding mechanism. We are privileged to have Dr. Brijesh Taksande, Professor and Head, Department of Pharmacology, Srimati Kishoritai Bhoyer, College of Pharmacy, Kamti, Nagpur, as our esteemed resource person for today. At this point, I will like to request Mr. Chetan Patil to kindly introduce our esteemed speaker. Over to you, Chetan.
Thank you, sir. Good afternoon, everyone. It is my great pleasure to welcome each one of you for today's faculty development program. We are truly privileged to have among us an inspiring academic leader and researcher who has significantly contributed to the field of pharmaceutical education and advanced pharmacological sciences. Allow me to introduce our distinguished speaker for today, Dr. Brijesh Taksande. Dr. Taksande's academic foundation is firmly rooted in Nagpur's own RTM Nagpur University, where he completed his bachelor's and master's in pharmacy, followed by PhD in pharmaceutical sciences. His doctoral research focuses on the pharmacological effects of agmatine on feeding behavior of rodents, reflecting his early sustained interest in neurosciences. With a rich and dynamic career spanning over 21 years, Dr. Taksande has held progressive roles at Srimati Kishoritai Bhoyar College of Pharmacy, Kamti, Nagpur. Starting as lecturer in 2003 and rising to the position of professor in 2017. Since December 2021, he has been serving as a Dean of Research and Accreditation, where he oversees academic excellence, research initiatives, and institutional development. His research is both deep and diverse, specializing in advanced techniques such as immunocytochemistry, stereotaxic, intracerebroventricular drug administration, and range of behavioral model for studying drug dependence, reward, and central nervous system disorders. The expertise has enabled him to conduct pioneers preclinical works on antidepressant, anxiolytics, antipsychotic, and anticonvulsants. As a mentor, Dr. Taksande has guided 2 PhD scholars, 80 MPharm students, and 15 bee farm projects, nurturing the next generation of pharmacists and researchers. His scholarly output is remarkable, with the 150 publications, most in respected international journals, along with nearly 200 national and international presentations. Beyond research and teaching, he actively takes part in the academic community by attending conferences, seminars, and workshops, improving his knowledge and contributing to pharmaceutical sciences. Today, we have the opportunity to learn from a leader who blend deep scientific knowledge with extensive teaching and administrative experience. It is my honor to invite him to share his insights with us. Please join me in extending a warm and respectful welcome to Dr. Brijesh Taksande. Over to you, sir.
Dr. Brijesh, hello.
Hello, sir. Am I audible?
Yes, yes, you are audible.
Hello.
I think we are not audible to you.
Aman, sir, am I audible?
Yes,
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 yes.
sir. You're audible, sir.
मनोज आपला आवाज नाही चालला त्यांना रे.
There might be problem from his side.
ओके.
मनोज सर, आवाज आ रही है आपको?
Yes, yes, sir. You're audible.
उनको आवाज आ रही है तो और ऑडिबल
Am I audible?
Yes, yes, sir.
हाँ मन.
तुझा आवाज क्लिअर येतोय. अं, चालेल ना. नाही नाही चालेल. तू just wait. He is joining with another instrument. Bridget sir. Manoj, he has joined.
Yes, sir. Yes, sir. I'm seeing it.
Okay.
Yeah.
Bridget sir, we are audible?
Yes, sir.
Okay, now it's okay.
Oh.
Yeah.
पहले चल
Okay, we are getting your presentation.
Okay, so can we start?
Yes, sir. Yes,
Yes, sir.
sir.
Yes, sir.
Okay, thank you very much, Dr. Aman, Dr. Manoj. It was technical glitch. I could not understand why I was not listening to you, even though I was audible to you, but there was maybe some kind of problem from our
Yeah,
end.
yeah.
It okay. Let's begin with the presentation. At this outset, I just want to thank Dr. C.D. Upasni sir, my good friend, just like a big brother, and my mentor as well in the pharmacology. And Dr. Aman, who is always being a good friend, not only as a researcher but also as a junior to me. And thank you very much for training my two James, Dr. Mayur Kale and Dr. Neetu Ankede, to the extent that they could grab the grant right now. So we are having a research grant fetched by Dr. Neetu Ankede and Dr. Mayur Kale at our laboratory, and credit almost goes to Dr. Amanup Gannawar. Thank you very much for this opportunity. So, to begin with this chapter, to begin with this session, writing effective research grant proposal uh, for for ICMR, I have intentionally chosen this ICMR funding research opportunities because uh, at present only this call is open for the new researcher, for the established researcher, and there is tough competition to grab the research funding opportunities from ANRF. That's why I will be focusing more on small grants specifically designed for the new researcher, and we'll be also focusing on different components of the research grants for the small grants, so that it will be having a win-win situation for everyone who is attending this particular session. And to begin with, I just want to have one disclaimer. Uh, Securing a research grant is a long, rigorous, and sometimes a first frustrating process that demands patience, continuous strategic efforts, and hard work. If you are interested in instant gratification and immediate rewards, you can consider participating in either Kaun Banega Crorepati, or a visit to any casino, or a purchasing a lottery ticket.
Correct.
Research funding is a rarely a game of chance. So, if you are considering that immediately after learning this, I will be having expertise to the level that I will be submitting a proposal to any funding agency and immediately I will get it. This is euphoria, just come out of it. It's a very long and rigorous process and involve a tremendous hard work. It requires tremendous hard work as well as continuous strategic efforts. Then and then one can have funding to his or her credit. So, what is the importance of the research and research grant in pharmaceutical sciences? As we are all aware that research is essential for the drug discovery and development process, as we are all the integral part of pharmaceutical sciences directly related with the healthcare maintenance of the society. It is our essential duty to have the new drug investigation so that it will be beneficial for the society at a large. So research grant, having a research grant and to continue with the research is almost essential for drug discovery and development process. It is also required to investigate the different disease trends and the risk factor by which human can suffer from. It is also responsible for the development of new treatment options for the incurable diseases. It can address the rising infections and trends and can afford the medical conditions. It can make the therapy affordable, as we can learn from COVID-19 era, or infectious are rising and alarming at an alarming speed. So it is essential duty to have the therapies affordable and accessible to all the society. It is also essential to monitor and evaluate treatment of outcomes and public health interventions, and also responsible for the foster collaboration and knowledge sharing in research and development. So ultimate aim of any research or research grant is to translate the research finding into clinical practice and to provide a concrete evidences so that policy can be framed at the government level or at the institutional level. It will be beneficial for the society at a large. So what advantage it will be providing to the institute? As you can see from many accreditation agency or assessment agency, if you get a funding from any government agency, it will be creating a good image or reputation of your particular institute at national or international level. It will be associated with the contribution in the development of new knowledge, concept, or any theory to the science. It is almost required or essential for accreditation and assessment by any agency like NAC, NBA, or even nowadays we are facing QCI infection. So accreditation and assessment agencies need to have the funding which is grabbed by any faculty member of its institution. Ultimately, it will be also associated with good NIRF ranking by HRD, Government of India. And most important thing, if you get one grant, ultimately more grant will be followed. It will be also associated with new publications, patents, other types of government funding, and also you will be getting funding from funding in the form of consultancy from any private firm or private pharmaceutical industry. So from institute point of view, there are many advantages, not only in the reputation, not only in the building of images at national or international level. Actually, it will be associated with the development of sophisticated laboratories. It will be also associated with the procurement of chemicals, procurement of sophisticated equipments, maybe recruitment of JRF, SRF would be the helping hands for the principal investigator. Now, what advantage does faculty have by all these things? So, like one can easily say, ki my life is very good. I'm in the academic from last 15-20 years. I'm getting 2 lakhs per month, and why should get I should get involved in the fetching or grabbing of research funding. So ultimately, it is associated with the employment security and stability. I used to tell my faculty members or my colleagues that whenever I get any grant, I say कि 3 साल का जॉब तो पक्का है. So almost it will be providing you employment security and stability. It will also enhance your academic performance indicator. You will be having the satisfaction for the contribution to the existing science. It will be ultimately related with the patents, maybe in the form of publication as an outcome. And it will be also providing you different avenues for the prospective research collaboration. So as far as faculty member are concerned, first and most important thing is that whenever you are bringing funding to institute, there will be sense of security, there will be sense of stability. Like unless and until it get completed, nobody can harm you, nobody can take away your job from you, right? This is my personal experience. It will be ultimately enhancing academic performance in the form of research, and would be giving you much more outcome in the form of patents and publication. But there are some misconceptions. right? When I talk with many faculty members, this is very interesting to know that many faculty member who are working from last 20 years, 25 years, they have this myth or perception in their mind that you can't get grants at the beginning of your career. First and most important, false claim. Second, grants are too complex for me or my team. And sometimes they used to say it's too late. They get good grants because they have been getting them for years. This is not true. Any faculty member at any age, at any time frame of his or her own career can get the funding from any funding agency, provided he comply with the guidelines, provided he should have developed the research profile up to the extent that would be eligible to fetch him funding, and the proposal has been written in a very well mannered. These are only two or three criteria which are ultimately responsible for getting a funding from any government agencies. So I will be discussing more about the funding which are now open as a call for the funding proposals. And mostly I will be dealing with ICMR research funding proposals or opportunities in this particular presentation. As I told you in the beginning, ANRF is not providing funding right now. There is no open call at this time point. So I will be not focusing on the research proposals related with the DST or ANRF or DHR. Only will be focusing more on the research opportunities available for the biological researchers, and that to be dedicated only to ICMR. So ICMR, that is Indian Council of Medical Research. The agenda aligned closely with the national health priorities, focusing on reducing India's disease burden across the communicable diseases, reproductive health, maternal and child health, nutritional disorder, and non-communicable diseases. They provide funding in four specific domains, especially discovery research, for example, generating proof of concept studies and preclinical validation. They also provide the research funding for development research, like creating interventions and improving the existing interventions for any particular disease or disorder. They are also providing funding to delivery research, for example, implementation of research to overcome barriers directly related with the clinical trial. So from starting phase 1 clinical trial to phase 3 clinical trial, you can get funding from ICMR. And they also provide funding to descriptive research. For example, epidemiological studies and observational studies are also considered for the funding in ICMR research funding proposals. So who can apply for it? Any researcher who is belonging to India, Indian scientist with regular employment, any faculty of medical colleges or research institute, having regular employment in university and R&D laboratories are eligible to apply. And as you can see from the figures, they have a huge funding for research in India, right? ICMR have changed its scenario from last 3 years, and now they are providing funding to every kind of research domain, to every kind of research opportunity, to all the researcher across the India without any discrimination, right? They will not be discriminating whether you are belonging to any private college, whether you are belonging to any research laboratories, whether you are belonging to any medical college. So despite your belonging, despite your affiliation, if your idea is good, if you have a significant problem in your hand, and if you have a significant solution to it, they are ready to provide funding to every researcher across the India. So there are three extramural research funding right now available for any researcher of the India. They are divided into three domains: small grants, which is up to 2 crores, maybe for 4-year maximum duration; intermediate grants from 2 crores to 8 crores. And they also provide funding for the development of Center for Advanced Research, The money is from 8 crore to 15 crores and total duration is for 5 years. Now, what is the purpose of small grants, intermediate grants, and Center for Advanced Research? As you can see from the presentation, the small grants are designed with the purpose to have the time-bound projects with specific measurable objectives aligned with the ICMR priorities. Whereas the purpose of intermediate research grant is mostly related to validation and evaluation of intervention with the proven concept must stems from well documented preliminary work. So if you are dealing with the small grants in the beginning, you are eligible for the intermediate grants after the completion of small grant research project. Whereas Center for Advanced Research designed with the purpose to provide the advanced research to a facility to experienced team, maybe single or multiple link project with the clear deliverables are are expected out of Center for Advanced Research, which is popularly called as a CAR scheme for the research, and it is expected that all three extramural research project would be associated with some form of outcome, some form of output in the form of publication, in the form of patents, in the form of direct deliverable for the benefits of the society, for the benefits of the patient, or establishment of a center where you can train the personnel, you can have the sustainable research program, and they are continuously monitored through their programs, through their monitoring system. So expected outcome in terms of small grants, it is expected that you will generate some preliminary data for the larger grants. Publications are expected out of a small grants, and there would be expectation from any new methodology development. So it is best suited for a new researcher, for a multidisciplinary project addressing priority disease and proposal building on a previous ICMR grants. Whereas intermediate grant are specially suitable for the clinical trials, or where you can expect the established researcher will be applying for the funding. The study may be multi-site or multi-centric. It will be giving funding for the clinical trials from phase 2 to phase 3, and also for the validation of any new technology generated out of your project. So what is expected out of intermediate grants, that it will be generating commercial product, following the patents, impactful publication, and influencing clinical practice. And car centers are expected to have the trained training of the personnel. It will also lead as a torch bearer for new research area, for development of new research area in very specific remote domain. And this is what is expected out of all these three grants. So roughly speaking, small grants are designed for a new researcher who want to establish themselves as a research scientist in academic or research laboratory. Intermediate grant is designed for the established researcher who have worked for research project, maybe for 2 or 3 research project in his earlier journey. And Center for Advanced Research would be providing a funding to a multi-centric research stream, and it is usually associated with a long run process. It will be associated with the establishment of a center which will be leading the country for a outcome of very specific remote research domain. So there are calls for first in world challenge by ICMR, and they are giving funding from 1 crore to 8 crores. So if you have proof of concept, you can get 1 crore. If you have a prototypes developed as a product, you can get 4 crores. And if you have a final product in your hand and you just want to check it through clinical trials, you can get up to 8 crore fundings. But this is very specific funding. You have to establish as a researcher, and you have to validate that whatever the problem you are submitting as a proposal, it has got first time in the world opportunity or first time world challenge. So nobody has done it before, or nobody has thought it. Nobody has given any consideration to that particular problem in that particular idea provided by you. So it is very remote funding. It is usually given to the established laboratories or established researcher who can think out of window and providing very novel, very first of its own kind, state of art innovation technology to existing problem of Indian generation. So what are the current funding interest of ICMR, where they will fund and where they will not? So these are the priorities area which are identified by the ICMR, and they have prioritized it, right, depending upon the prevalence, depending upon the epidemiological data. They have certainly came out with some priority area, and even you can look at the funding uh, proposals which are submitted by the investigator, and you can see from the results that usually funding distributed amongst these priority areas only. Okay, so they have three main domain where funding can be submitted, funding proposal can be submitted. First and most important is One Health, which is integrated human animal environmental approach for the health. Second most important area for the ICMR funding is antimicrobial resistance. This is very challenging situation for all of us, that most of the antibiotics which were working earlier nicely have stopped giving positive result for the treatment of microbial infection. So antimicrobial resistance is one of the hot cake and priority area for all infections. Tuberculosis, vector-borne diseases, respiratory infections, HIV, AIDS, their prevention, treatments, and co-infections are considered as considered as a domain which are coming under the communicable disease, and considered as a priority area for funding by ICMR. Now, what included in the non-communicable diseases? The first and most important non-communicable disease is cancer. As you know, there is a high spread of cancer. There is a like tremendous increase in the number of cases diagnosed with the cancer nowadays. So most important funding opportunity uh, to get the funding is to apply in the cancer domain. Second to it is a cardiovascular diseases, diabetes mellitus, mental health, chronic kidney diseases, and chronic respiratory diseases are also considered for the funding by ICMR in its priority area. Apart from this, reproductive and child health, for example, anemia, micronutrient deficiencies, iron deficiency, vitamin D deficiency, B12, folate deficiency, all these can be considered as a priority area by ICMR. Childhood malnutrition, neonatal mortality, for example, preterm birth, low birth weight, sepsis, early childhood development, for example, neurodevelopmental outcome in small newborns, uh, polycystic ovary syndrome, endometriosis, reproductive health disorder in women, and some genetic or congenital abnormalities, inherited diseases are also considered by ICMR for funding. The most cross-cutting priorities nowadays dedicated to aging and disabilities. They are also funding tremendously for oral health, climate change, and its impact on the health outcome. So all in all, you can consider the health priority of the India. You can consider the prevalence of a disease across the Indian state, and depending upon the epidemiological data, depending upon the prevalence, they have given the priority to particular diseases. And if you could submit your proposal in this particular area, the chances are very high कि it will be considered for the funding. So this is given by ICMR in its statement कि they are the priority area of ICMR for funding. And if you could submit your proposal in this particular area, the chances are very high that you will be getting your proposal funded by ICMR. Now, this is the trends of ICMR to which they have funded in last 2 years. As you can see from this graph, 20.6% of the funding goes to cancer, or total project, when we distributed into different domains, mostly cancer received the funding up to 20%, followed by infectious disease, which was about 19.7%, then up to 11% neurodegenerative disorders, neurological disorders. They also provided some form of funding to diagnostic up to 7%, maternal and child health up to 7%, drug delivery systems, development of nanomedicines, ophthalmology, gastrointestinal and liver disorders, and other rare categories also received the funding from ICMR. But as you can see from this graph, funding mainly sanctions to cancer and infectious disease, followed by neurodegenerative and metabolic disorder, reflecting India's major disease burden with moderate support for diagnostic, maternal child health, and translational technology, right? So if you're working in any of this area, there is high chance there is a tremendous opportunity for new researcher to develop a proposal in this particular area, because you can see from the trends, you can see from the pattern that most of the funding is received by the topmost trending disease of today's area, that is cancer, followed by infectious diseases, followed by neurological health, followed by metabolic disorders. So you can consider applying for the research proposal, taking your research expertise in this particular domain and applying for the ICMR research grant. Now, how this process will go on? What is the pathway for proposal generation to its submission? How will will formulate a hypothesis? How will initiate with the identification of a research problem? How will review the different literature available, and how will submit the proposal, right? So this is a chronic process. As I told you in the beginning of my lecture, that this is a chronic process. Like, if I'm saying ki call is open, and you just search on the internet, or on the Google, or if you ask to chat GPT, you will find some ideas, or you will get some clues about what could be the areas which can be targeted for funding proposal, and how to submit the most promising research proposal to ICMR, right? So it usually begin from identification of a research problem, right? So research problem is nothing but the disease. Keep in mind, disease is the only triggering factor for any drug discovery process, right? If you have anything in mind, like yeah, I'm working on any phytoconstituents or any phytochemicals, I'm working on a novel drug delivery system, and I will be focusing its application to any of the disease or any of the disorder randomly. You're wrong. First and most important triggering factor for any research proposal is a identification of a proper diseases, right? Once you identify the particular disease, you will find what are the current therapies available for it, what are the drawbacks of those current available therapies, where the therapies are getting failed. Now, if you come to the conclusion, these are the current available therapies and they are getting failed, you will be again going to the internet or going to books for the review of literature. So, if you identify a particular disease, it is expected that you know everything about it, right? What's exactly involved in its pathogenesis, what are the different triggering factor for that particular disease or disorder, what are the different pharmacological domains by which we can treat, or we can reverse the disorder, if not completely treated, can we minimize the symptoms, or can we subject it to mitigation. So basic idea is to know everything about your research problem or everything about your target disease condition. So after coming to the basics of the research problem, go for a comprehensive review of literature. Right. So you will be searching on internet, adding the keywords by that particular disease name or by that particular disease condition, and collect the data which is generated in last 3 or 4 years. Right. Identify the gaps and consider whether you have any solution for it. Right. This solution can be anything. It can be novel drug delivery system, it can be any new drug candidate, it can be any phytoconstituent. But to identify a gap, following understanding of any research problem is the key. Right. Unless and until you understand the disease completely, you will not in a position to find out the research gap in that particular domain. Right. So once you have identified the research gap and you have identified the possible solution for it, you can formulate the hypothesis and objective of your research proposal. Okay. So you have formulated the hypothesis, you have formulated your research question, you have also formulate or design your different objectives. Right. It should not be more than three or four. Okay. So now you have identified different research objectives, and depending upon your laboratory conditions, depending upon the facilities available at your institute, depending upon your own expertise, next stage is to identify who would be my co-investigator or collaborator. Keep in mind, one co-investigator from your own institute is mandatory, right? So even if you hit all of them, consider one co-investigator from your own institute. This is mandatory thing. Like, and then you can search for different research collaborator who are working in the same area and whose collaboration will be actually adding value to my research proposal submission. Right. So this is not very critical process. You usually know who are the principal investigator working in that particular research area and how they would be helpful for my proposal submission, and how their involvement in my proposal would be adding benefit or would be adding value to my proposal submission to ICMR. After this, there would be interaction, there would be communication, there would be different meetings, and following those meetings, there is a time for the development of a protocol, right? Protocol designing is again very much important. Even if you have a very significant world changing idea, disease changing idea, even if you have a very concrete solution for any disease condition, and if you haven't designed your protocol correctly, it would be subjected to desk rejection. Believe me, even if you have a great idea, even if you are working on very novel solution related to any disease problem, but if it is not associated with proper protocol designing, it will be associated with the straightway rejection. Then after designing a experimental protocol, you will be looking at your expertise, you will be looking at your conditions, feasibilities for the completion of that project, and then you will be looking for an additional requirement. Right. So what additional requirement are underway, whether they are in the form of chemicals, whether they are in the form of sophisticated equipments, whether you need a provision of any JRF because you will be requiring hands for the completion of the project. And after everything, there is a time for the collection of a preliminary data. Right. If you're already working on any concept, any disease state, most of the time you have a preliminary data in your hand. It can be considered as a proof of concept for the development of any prototype, right? This is very important to generate or to arrange the preliminary data in a very sequential manner so that it will be building a confidence to the reviewer of ICMR funding agencies that this particular team, this particular principal investigator is currently working in this particular domain, particular area, and he has already worked in his early career, has published papers related to this particular research area, and there is no problem in giving him funding for the extension of his research, right? Then after this, there is a time for the writing a proposal as per the formats and guidelines. There is a time for the for a critical consideration regarding budget justification. So everything like it is usually given in the formats, or usually you have to follow the guidelines. It is just like a process of a paper submission or research paper submission, right? You just have to follow the guidelines, you just have to follow the formats, and critically you need to see whether your budget is appropriate and you have given justification for all your budget heads or not. And after uh, writing it, edit it, proofread it, read it 2, 3, 4, 5 times, 10 times, and then successful submission of research proposal. So these are all the different steps involved in the submission of your research proposal to ICMR funding agencies, right? But the first and most important thing is to identify a research problem. Okay. So how your research problem would be defined? How one can jump directly on any research problem, right? So there are some consideration, there are some suggestions from my point of view. If you are working on the problem which are very related or associated with Indian scenario, chances are very high कि you will be getting funding. For example, if I ask you a question, how many new drugs are developed for the treatment of tuberculosis, leprosy, or for leishmaniasis or kala azar in last 10 years or 15 years? You will not even recollect or recall any new drug candidate for the treatment of tuberculosis or leprosy discovered in last 10 years. The problem could be associated with its low profit potential because these are the diseases which are usually associated with low socioeconomic class, and even the private organization, private pharmaceutical industries do not want to uh, invest the money which are associated with the low profit margin. On the other hand, you can recall the drug candidate which have been which are available in the last few years for the treatment of cancer, for the treatment of lifestyle disorders like diabetes or cardiovascular complications. There are number of drugs available. The reason is very simple, because they are giving a high profit potential, and usually they are associated with the like urban population or high class society, right? So investment is also subjected to the profit. If any private pharmaceutical industries are investing, they're expecting high profit out of it, right? But ICMR do not discriminate between these two factors. They will be giving the priority to TB, they will be giving a priority to leprosy. Also, they will be giving a priority to cancer or any neurological disorder or any cardiovascular disorder or any metabolic complications without any bias, provided your proposal should be in order, should be giving a nice solution to the existing problem, and should be written as per the formats and guidelines which are given by the ICMR, right? Despite all these things, we can also work on the problems which are very near to our society, like faced by our day to day, which are faced by the society at very large. For example, alcoholism is a growing concern. So is the case with smoking. Smoking is not only associated with the addiction, but also associated with so many types of the cancer, right? So alcoholism, smoking, or drug addiction is considered as a new area which can be targeted for the submission of proposal to any funding agencies, right? So without any discrimination, ICMR will be funding or will be giving opportunity to all the research proposal if it is written nicely with appropriate guidelines, and if you are providing a novel solution for any existing problem, they are happy to provide you funding. So after identification of a disease, there is need to identify a drug target, right? So drug target is a specific micromolecules, maybe protein or any biological system, maybe enzyme, which the drug will interact with, right? Sometimes this can happen through incidental observation, right? So while going through review of literature, this is my personal suggestions to all the participants available here. Try to find out what are the clinical trials which are directly establishing the correlationship of a disease to the particular drug molecules or particular endogenous molecule, right? There can be overexpression of the protein, there can be down regulation of the protein, there can be dysregulation, maybe increase or maybe decrease of any indigenous molecule, and this would be giving you some clues. It would be giving you some opportunity to establish dysregulation of that particular endogenous molecule to the progression of particular pathogenic mechanism for triggering of that particular diseases or not, right? So after identification of a disease condition, it is very important to identify how it affect the physiological systems of our body, how many endogenous biological molecules are there which are overexpressed, which are down expressed, which are down regulated, which are dysregulated, or maybe sometime there is increase or there may be decrease in their quantity at various level, maybe at the level of periphery, maybe at the level of CNS, maybe at the level of different areas of the brain, right? So have a critical look at the different literatures, different research articles available to state this particular correlation, association of a disease condition to the dysregulation of a biological molecules. This would be the key pathway by which we can identify a new drug target, even we can identify a new drug candidate for the existing problems. So these are some suggestions where you can target the development of a drug candidates, right? So it can be any neurotransmitter, for example, serotonin, acetylcholine, norepinephrine, or dopamine. You can see there is a direct correlation between their dysregulation to the disease conditions, right? I will be focusing more on the biological molecules which are associated with CNS. uh, this is just a suggestive, right? You can go to any molecules which are available in the periphery, which is hormone, which is any other neuromodulator, and you can establish its correlationship with the progression or even the triggering of a particular disease, right? These are my suggestions, right? Where the drug discovery or development process can begin targeting this endogenous molecule, right? So after neurotransmitter, there are some neuromodulators like BDNF or nitric oxide, which can be targeted for a drug discovery process. There are some hormones uh, like insulin and oxytocin. You know it for sure that insulin has a very important role in the pathogenesis of diabetes. Oxytocin again associated with like labor pain and other issues. But nowadays there is a change, trending opportunities to work on the application of insulin for the inhibition of the memory impairment. Oxytocin is again a particular hormone which can be established as a particular molecule which can stabilize your emotions, right? Apart from this, there are some neuropeptides like NPY, NPS, and relaxin, which can be again targeted for the development of a lifestyle disorders which are associated with the exposure of particular chronic stressful conditions. There are some neurosteroids like allopregnanolone can also be targeted for the development of a drug discovery process. Amino acids like L-arginine, essential amino acid, semi-essential amino acids can again be targeted for the development of new drug discovery processes for any disease conditions. And there are some biogenic amines. For example, agmatine, on which we are working from last 20 years, can again be targeted for the development of a new drug candidate targeting particular disease conditions. So these are all my suggestions. I have been working on these endogenous molecule from last 20 years, and I can suggest you that if you find out the concrete correlationship between these neurotransmitters with the pathogenesis or with the progression of a particular disease condition, you will be having a concrete evidences to correlate that particular disease with this particular biomolecule and can have the discovery as a drug candidate. Okay. Then like you have identified the disease, you have also identified the possible or probable endogenous molecule to work on a particular drug, particular candidates. How to continue with the review of literature, right? So review of literature is a continuous process. You have identified two things. One is disease conditions have been identified, that you have to work on a particular disease condition. You have also identified what could be the probable biological molecules which can be correlated with the progression of a diseases, and if I can restore the quantity of that particular neurotransmitter, neuromodulator, I can certainly have the inhibition of that particular disease condition, right? So first and most important stage in this at this time frame is to review the basic concept and theory. Again, you have to go to your books to find out what exactly is the pathogenesis of that particular disease. What are the missing link? Why the solution is not coming out? What are the existing limitations for a particular pharmacotherapy and how we can overcome it, right? Then you have to refine it through comprehensive literature review. I suggest you to focus mostly on the data which is available in last 5 years. So for example, if you have identified two condition, one is disease and second is biological molecule, by adding these two keywords on Google, you will get 1,000 article, right? If you cut it to last 5 years, you will get 100 articles, right? Now again, you will have to cut it down to only research articles, right? So 100 articles would be comprising of case studies, would be comprising of a research article, would be also comprising of a review articles. Just ignore them. Only focus on the typical 7 to 8 highly specific research article, right? Conduct independent literature survey. Don't believe on anyone, like don't depend on anyone, right? If you have identified your disease, if you have identified the probable drug candidate, go and conduct independent literature survey. What could be the correlationship existing between these two domain, right? After specifying a particular research area, particular solution for it, you will be defining a research problem. It may be research question or it may be research hypothesis, right? Believe me, your research question, your research hypothesis will be directly dependent upon these 7 8 articles which are identified by you personally at this particular time frame, right? So what is the different process to identify the different research gaps? how we can find out the solution for it. If you divide it into three columns: disease, drug, and solution, just find out the answer for this. What is known about that particular disease, right? So what we understand, what is the background of that particular disease, what is the etiopathogenesis, and is there any need to have the new solution to new therapy for it. Then again you will have to answer what are the drawbacks of current pharmacotherapy available for its treatment, right? Then what is unknown about it? You know it, but what is not known to you, right? And third and most important thing is that if you have identified a possible drug candidate, how you're going to provide a novelty to it by addressing the problem, by giving a solution in the form of novelty, right? So first and most important thing is to find out what is known to particular disease thing, what is unknown about it that is associated with the drawback of a current therapy, and how your drug candidate or how your new molecule which you have identified by your own efforts, by your own literature survey, going to find out solution for it, right? We'll discuss this in next few slides. So first and most important thing which need to be considered is what is the knowledge available in that particular domain, right? So what is the knowledge, which is a deep understanding of a field, principles, and established fact. We can't change it, right? But information will keep on changing. Okay. For example, space travels causes cognitive, muscular, and metabolic impairment in astronauts. This is a knowledge. It will not change because experience has been written in the books, and they have available, they they are available in the form of concrete evidences, right? It is not going to change because space travel usually expose you to radiation, to microgravity, ultimately associated with the like speed up of your aging, may be associated with cognitive, muscular, and metabolic impairment in astronauts. So what is information available? Information is that particular thing which will be keep on changing, right? As far as this fact is known to us as a scientist in pharmacology, exercise has got its own beneficial effects. So exercise releases, this is information not exactly established. Exercise releases exerkines like irisin, BDNF, and can have the direct impact uh, on the physiological systems of the body, and can be providing the solution to the cognitive impairment and metabolic impairment, right? So see, there are two things. One, you're discussing a problem which is associated with the established information. Second, you're providing a solution by depending upon the information, right? Third and most important thing is that whenever you are providing a solution to a particular problem, let's let's judge it properly whether that problem is a uh, properly addressed by your solution or not, right? Then you will be coming to the identification of a gap, right? So I know exercise mimetics molecule like irisin or BDNF can be associated with a treatment of a cognitive impairment or muscular or metabolic complications in astronaut exposed to space exploration, but I cannot give them directly to the patient because they are not the drug candidate, right? So what is the problem available with them? They degrade rapidly and cannot be easily translated into therapeutics. So this is the information I know for sure. There are some candidates which can inhibit the cognitive and metabolic impairment in the astronauts, but I cannot treat them as a drug candidate because of its rapid degradation and having drawback as a translational potential. So what is my solution? I can incorporate these exerkines or exerkines mimetic molecules into one drug delivery system known as exosomes, and we can have the solution that to that particular problem. So novelty exists in a way that exosomes based delivery of exerkines can be the solution for a space exploration. It can be developed as a space medicine for the astronauts, right? So this is the way how we have to identify the problem, how we have to identify the gap, and how we have to develop novelty so that it will be properly justified in the form of a solution, so that it will be easily acceptable by the reviewer of a funding agency, so that it will create a confidence in them, and ultimately responsible for getting the funding proposal sanctioned. Now, how to formulate the hypothesis? This is again a very interesting question. Ki formulation of a hypothesis is depending upon your strong literature review. Unless and until you know everything about your problem, you cannot generate a good hypothesis. So, what is hypothesis? It's a predictive statement about a possible outcome of a research study based on a strong scientific data. It should be very specific, it should be very clear, and most importantly, it should be testable proposition or should be novel and convincing, right? So exosomes based delivery of irisin may mitigate microgravity induced muscle and cognitive impairment by enhancing neuromuscular resilience is my hypothesis in this particular research domain, right? It can be anything, it can be any possible outcome, but it should be very novel and it should be very very convincing to the reviewer of funding agencies. So how to design a protocol? This is again very important. As I told you in the beginning, key protocol designing is very important, right? It should cover everything, almost everything required to satisfy your objective. If your objective is claiming something and if it is not satisfied by your designing of a protocol, ultimately it will lose your chances to get your proposal sanctioned. So plan the study with clarity and reproducibility in mind. Develop standard operating procedure with detailed parameter. Even if there is a word limit, but try to give maximum detail about the procedures which are involved in your methodology. Ensure compliance and validate animal model. This is again very important. We write everything, but we forget to write कि my proposal would be subjected to approval by Institutional Animal Ethics Committee as per the guidelines of CCSE. This statement is just a statement, but very very important from regulatory compliance point of view, right? So we need to take care of everything, whether your proposal would be subjected to critical scrutiny at the institute level and will be following all the guidelines given by Government of India or not. Try to use the established behavioral protocols, and you should be very sure about what could be the endpoints of your protocol, right? So you should explain everything. What would be the my protocols and what is expected out of it? What could be the end point of my protocol? Try to conduct or try to incorporate integrated pharmacokinetics and toxicology assessment. See, we are uh, providing solution for any disease condition where we are proposing any new drug candidates or new drug entity to a particular problem. It is very important that it should at least satisfy the criteria of phase 0 or phase 1 clinical trial. And that's why if you conduct the animal experiment initially, it will be it will be beneficial for you to get your project sanctioned. Try to incorporate biochemistry, histopathology, and immunocytochemistry and their critical analysis. This is again very important, and the question would be asked by any candidates. I don't have any facility for histopath or immunocytochemistry at my laboratory. Even I do not have any auto analyzer at my lab, so that I can perform the biochemistry of that particular disease condition. But you can explore the option, you can resource it, but try to incorporate all these assessment criteria, all this protocol requirement in your proposal. Also, write very properly how you are going to analyze and interpret the data. This is again very important. Statistical analysis, biostatistics is one of the important parameter of your methodological section, right? Again, how you are drawing the conclusion and how you are going to report the finding. These are all essential component of your protocol designing, right? So uh, there should be clarity, there should be reproducibility, there should be use of established behavioral protocol, there should be compliance to the regulatory body, there should be initial experimentation carried out at the laboratory level, and it should be completed in all the sense so that it will be providing confidence to the examiners of your proposal so that proposal will get sanctioned. As you are aware that we are all pharmacologists, right? Access face validity to phenotypic similarity to human disease. If you're using experimental animals protocol, try to find out if you're using a particular disease condition animal model, whether it has got face validity, whether it has got a construct validity, or whether it is providing any predict validity or not, right? So there are some examples to provide a good high validity models. If you're using a transgenic or knockout knock-in animals, it is creating a positive impacts on the reviewer. Environmental models, pharmacological models which are directly associated with the disease conditions are again very useful to build a confidence in your proposal examination. So don't use any other animal model which are available at your laboratory, right? Try to use only those animal models which are satisfying face validity, construct validity, and predict validity. Then and then it will be subjected to a proper evaluation and will be having high chances for the approval. Otherwise, it would be straightway rejected. So these are some schematic representation of the protocol, right? So it is better to give the schematic representation of your all experimental procedures, protocols, what would be your behavioral phenotype, what would be your biochemistry, what would be your immunohistochemistry protocols, what are different studies you're going to perform at molecular level, at behavioral level, at a cellular level, or at a genetic level, whatever it may be. Always try to provide the data as a schematic representation, right? So this is just one diagrammatic representation for the scheme of the protocol designing. Write in bold letter your title, how you will begin with your protocol, what are the morphological assessment, what exactly you are going to perform in the animal experimentation, everything it should cover, right? So if they do not get time, just by looking at the diagrams, they will have an idea that yes, everything is covered, and that's why it is ultimately building confidence in your research proposal. So this is second example how we can design the experimental protocol, and after experimental protocol just give it in the schematic diagram, right? So everything should be covered, what exactly you're going to perform and what is expected out of it through diagrammatic presentation. This is third schematic representation of your uh, of your protocol. This can be given in the proposal itself. Sometimes we forget the most important issues to mention in the proposal. There is a tremendous importance of inclusion of sham control vehicle control group in the experimental design. Always use positive control or a standard drugs wherever applicable. Always incorporate that you are going to have the baseline measurement of all the pathological parameters before intervention. Why this is important? Because we cannot predict the intervention's outcome, and it is better to have the preliminary data in your hand what exactly your drug treatment is going to do with the animal experimentation protocol, right? Randomize animals into different group. This is again very important. Some issues which are very important again to mention the details of methodology. For example, even if you're performing a very routine test like locomotor activity, cognitive assessment, anxiety assessment, depression, you're using any of the parameters. For example, you're using open field test, or you're using actophotometer, or you're using Morris Water maze for the evaluation of a cognition. Mention every detail in your proposal. So is the case with the monitoring of physiological data. How you are going to weigh the animal, how you are going to check the temperature, how you are going to check the food consumption, water intake of animals should be mentioned very clearly in the proposal. So is the case with biochemical data. How you are going to assay the enzymatic analysis, how you're going to analyze the oxidative stress markers, how you're going to analyze the neurotransmitter level, which methods will be applicable. You have to mention it very critically, very clearly. So is the case with molecular and histological data. How you're going to determine the protein expression, immunohistochemistry, or what microscopic techniques you're going to use. So everything should be given in a very detail, right? The most important parameter one should not forget here, whenever you are giving a method for a particular assessment, write a proper reference to it, a true reference. Giving a wrong reference is a suicidal behavior. If you are giving a wrong reference to any established method, there are very high chance that it will be straightway rejected. So there should not be any compromise with the reference as far as your methodology is concerned. It should be very correct, it should be very timely, and it should be very established fact that everyone working in that particular area know about it, right? So take care of these things while writing your methodology, because unless and until your methodology is supported with your objective, it would not be giving you a outcome in the form of result, or outcome in the form of the data which is generated by your methodological procedures, right? So to follow the proper methods, proper timing, proper animals, genders, sex ratio, you have to justify everything while designing your protocol. Apart from this, there are some confounding factors which plays very important role in the designing of experimental protocol, and we usually forget it, right? So what are the biological factors? For example, age, sex, genetic background, hormonal cycles. If you are using the female animals in the experimental protocol, you should take care of it. So is the case with environmental conditions like housing density, cage enrichment, temperature, noise, light dark cycles. Also mention very critically what would be the diet composition, water quality, feeding schedule, injections accuracy, drug formulations, handling stress, and observer bias, subjective scoring of the behavior. So you have to mention everything. every detail in the methodological sections, how you're going to observe the behavioral phenotypes, how you're going to monitor the different conditions during the methodological processes. So everything should be written very, very clearly to get your proposal sanctioned by any funding agencies. So if you have any data in your hand, which can be considered as a prototype, which can be considered as a proof of concept of a data, organize it properly, right? So the data of primary importance should be given a first chance, then to be followed by a secondary data. Organize it with proper subheadings, logical sequence, and also gives statistical analysis, right? This is again very important. It actually provide to build the confidence in reviewers, so that reviewer will feel confident about your submission, right? If you have data in your hand, preliminary coming out of preliminary experiment, it will be uh, providing confidence in your proposal. So these are some suggestions how we can draft a good proposal. What are do's and don'ts of the language and style? Use simple, clear language. Avoid jargons and complex sentences. Follow word limit strictly. Exceeding may lead to rejection. Use active voice and present tense or future tense. Proofread multiple times. Avoid spelling, grammar, formatting errors. This will be associated with straightway rejection of the proposal. If they find some spelling mistakes or grammatical errors or formatting errors, they will not take it to the next level. Use structured, abstract, and clear subheadings. Don't use vague statement. Don't include personally identifiable information in part A, and don't use abbreviation without defining them first. Your objective should be very clear, very feasible, quantifiable, and it should be time bound, right? Demonstrating preliminary work is of tremendous importance. Like, mention what key findings were there, what was the sample size of the animals. Also include what is the profile of your team members. And this is very important to understand that principal investigator alone will be responsible for all the processes of uh, funding proposals. He will be responsible for recruitment of the JRF. He will be responsible for procurement of chemicals, animals, even the sophisticated equipments. His profile will be added on by the co-investigator. So use the profile of the co-investigator, use their expertise wisely for the completion of the experimental protocol, right? So what are the key factor for the consideration of the proposals? Significance of the problem, that is need of this particular solution, plays a very important role, right? For this case, whether you have identified the right problem and whether you are providing a right solution to that problem is the key. Novelty and originality, again, very, very important, right? As I told you, novelty plays very, very important role in separating your proposal from all the available proposals. Track record of the investigator again plays a very important role. His expertise, experience, his publications, his citations, his history of handling the research project earlier. Writing the finance or mentioning the right amount is very important. You cannot claim randomly what would be your budget, what would be your budget for animals, what would be budget for chemicals, what would be budget for your sophisticated equipments. Preliminary findings, pilot data if hand, again will be adding value to your proposal. Again, there is one more thing, like approach. If the plan fails, what are the alternatives available with the investigator? So if you clearly mention, if this is not happen, like if this is not goes as per the protocol, what are the alternatives available with the principal investigator to find out the solution? Scientific research environment at the host institute again plays very important role in the sanctions of the project, and ultimately overall impact. If you are submitting a proposal to any funding agencies, it should be fulfilling all the criteria given by that particular funding agency. It should be complete in the all sense. It should be novel, it should be original, and most importantly, associated or justified by the expertise and experience of the principal investigator and his team. Investigator profile, PI experience, expertise, and qualification plays very, very important role. You can't say this, कि I'm just a postgraduate and I want funding. Minimum PhD is a qualification required by any funding agencies. Publications, completed or ongoing project, more publication in the international journals like Scopus, Web of Science, more citation will help the researcher or faculty members to fetch the funding grants. The reviewer will be considering if you are at the early stage or new investigator, they do have appropriate experience or training. And if you are established researcher, then whether they have demonstrated an ongoing record of accomplishment and how advanced in their field. So in terms of publication or in terms of patents, they can look at your citations, they can look at your publication history, and they can justify whether you are a proper principal investigator to provide the funding with or not. So this is very, very important. Even if you have a good problem, even if you are having a novel solution for it, and if your expertise, your qualification, and your experience is not supporting it, there are remote chances that you will be getting funded by any agency. So this is part A, which is given in the ICMR guidelines as such. What it include? It include title. Uh, 25 is a word limit. It should be very specific, concise, descriptive, and informative. After that, they will be asking you to write a summary. And there is a structural subheadings. For example, rational gaps, novelty, objective methods, and expected outcomes. And then you have to select the priority area, followed by keywords and abbreviations. Then there is a problem statement. You have to write it in the 500 words. State currently available information to present the problem adequately with the context. Second is rational. How research question addresses critical barriers, relevance to local, national, international context with proper referencing or proper bibliography. Then what is your hypothesis? What is your research question? Right. One need to understand here what to be written in the problem statement, what to be written in the rational, and what to be written in the hypothesis or research question. Problem statement usually associated with the disease itself. You have to write everything what is known to the scientific community regarding that particular disease in a very structured format. The word limit is given. Rational is nothing but a solution. It may be any biological molecule, it may be phytoconstituent, it may be novel drug delivery system, but you have to provide a rational how this solution is going to address the critical problem which you have already mentioned in the problem statement. And depending upon these two domain, how you are framing your hypothesis and research questions. Right. Now, after completion of your hypothesis and research questions, you have to write typical objectives, not more than 3 or 4. Study design, we already discussed. It should be very, very clear. You have to write what would be the study area, what would be the sample size, and what would be the sampling strategy, project implementation plan, design of statistical analysis, everything. You have to mention everything in methodology. What are your outcomes and future plans, and timelines and support. So you have to give a graphical presentation how you're going to complete your project within timelines. And then there is a part B. What is to be written in type part B? Details of preliminary work done by principal investigator, including source of funding, up to 1000 word. This is actually referred as a proof of concept. Okay. Skill and experience of the research team highlights only salient features along with 5 relevant publications that provide confidence to the reviewer that the team can implement the project with quality. Third, institutional support and facility. Share a brief note on interdepartmental or interinstitutional collaboration needed for a study implementation. Do mention the role and responsibility of PI and co-PI. What are the laboratory facility available? For example, if you have animal house, instrument labs, different equipments which are necessary for the completion of that particular project, mention it. Additional supplementation or data available, you have to give it in a different file, right? So this is to be included in the part B. The most important component of part B is the budget component. So you have to give a proper justification why you need staff or manpower. So depending upon their recruitment, research associate, JRF, SRF, lab technician, data entry operator, there is a there are guidelines for their salary. Equipment, justify the need, uses, alternative, and how you are going to dispose it. Contingency, recurring, non-recurring, provide year-wise background, include consumables, lab supplies, reagents, printing, communications, or if you're purchasing any books out of it. So write clearly with the justification why you need a contingency. And additionally, they are giving a travel allowance. ICMR in these small grants are providing the provision of a travel grant for international travel, right? So up to 4 lakhs, you can ask for. They will be providing a funding for a international travel support. And overhead charges. About 3% of a recurring budget, you can claim the overhead charges, and which will be directly going to the institutional fund, right? You have to give justification for everything, right? Submit year-wise breakdown, include all the quotations, preferably through government of India's marketing websites. Obtain 3 quotations if it is more than 1 lakhs for equipments. Budget modification may be requested during the review. Unused fund must be refunded if project is discontinued. Okay. And after that, there is a requirement of signatures by principal investigator, account officer, and head of the institution with the stamp, right? So this need to be provided separately. So what exactly happens at the submission level, and how it will be associated with the approval of the proposal, right? So stage 1 would be initial screening. It would be taken place at a ICMR headquarters. And if your proposal is in the order, if it's in the good format, it will be subjected to independent expert review, right? So two independent expert review will examine your proposal, and they will be giving scoring depending upon the rationale, what would be the impact, what would be the innovation, methodology, and feasibility of doing the proposal or not, right? Then there would be stage 3, which involved project selection committee. It will be comprises of senior scientists and clinician, representative from ICMR governing bodies, and expert from the priority disease area. So they will review score compilation, committee review scores and commit from two independent expert, and then scientific discussion, ultimately associated with final scoring, and proposal will be ranked by score with each priority area. And if you're amongst the topmost in that particular area, it would be communicated to you regarding your selection, right? Sometime clarification may be requested from PI for any additional information or any modification in the protocol. Sometime there would be requirement of clarification as far as budget is concerned. Maybe PI is asked to revise the budget, and there are always suggestions for the improvement. Okay, so top rank proposal in each priority area will be funded, right? And if you're qualifying all these, you will be getting funding from the government agencies. So background, so this is the maximum score for any review of proposal. 20 marks are clearly given to the background and rationale of the project. Is it likely to solve the priority problem? What would be the possible impact? Is it likely to have the impact on health outcome? 20 marks are given. Novelty, innovation. Is the study developing or testing a new idea? 15 marks are there. Clear cut, correct methodology supported by good references are given 30 marks. So as I'm telling you from the beginning, methodology plays a very important role. Even if your idea is good, even if it is a novel, innovative solution, if it is not supported by the proper methodology, it would be straightway rejected. And last point that need to consider is implementation strategy, milestone to be achieved. So whether it is feasible, whether it is time bound, and out of this scoring, you will be considered for a funding agencies. So these are the timelines. 29th December, there was opening of a call, and it is open up to 16th March 2026 up to 5 p.m. So these are some points to be considered. You can submit the proposal in both the domain, small as well as intermediate. But keep in mind, if you are submitting a similar proposal, only your small grant proposal would be evaluated by a government agencies or ICMR. The PI must not have more than 5 ongoing research proposal funded by ICMR or sum of grant amount more than 25 crores. ICMR Scientists Institute are not eligible to apply in this call, but they can be named as a co-investigator. And at least one co-investigator should be from the parent institute. So this is the process for the submission. Kindly ensure that all the sections are adequately filled and necessary details. Inclusion of at least one co-PI from the PI institute is mandatory. And these are some guidelines that PR are advised to submit proposal well ahead in the time. So what are the strategies to get your project funded by ICMR? Align with the national priorities, maintain scientific rigor, build strong team, co-investigator and collaborators are necessary, demonstrate clear impact, ensure feasibility, and follow the guidelines precisely, right? You have to follow the guidelines so that it will not be subjected to a desk rejection. So this is all about the proposal from beginning, from identification of a research problem to identification of a solution, how to identify the gaps, and how to provide the innovative solution to the existing problem, and what are the different processes, which proposals are having the priority by ICMR guidelines, what are the different components of their statements, components of their drafting formats we have discussed. Now I'm just within 2 minutes I'm sharing what is our contribution in the pharmaceutical sciences. So at SKB COP, we are working on the understanding of the neural mechanism responsible for various diseases, and neuropharmacology emerge as a frontline area. There are some research components, for example, standardization and sharpening of skill in the range of neurobehavioral paradigms. We employ stereotactic surgery for intracranial drug administration. We also have neurochemical analysis in discrete brain areas. This is all supported by application of immunohistochemistry to visualize the neuronal function and its connectivity. I'm proud to tell you that we are working on this molecule, agmatin, from last 20 years. Only one molecule from last 20 years, and it has given us everything. Not only the project which are funded by ICMR, DST, DBT or DHR. Most of our MPharm students have worked for their thesis on this molecule. Most of our PhD candidates are working on the agmatin only for their PhD, and it has not given us only funding, it has also given us identity. The lab which is working on agmatin, a research molecule, and it is a few laboratory amongst the different 5 or 6 laboratories in the world who are working on the agmatin molecule. This is our journey, and this is the outcome. We have started exploring this molecule in 2005-6, came out with first publication correlating it with the depression, pathogenesis of depression. Then we have identified its application in various psychiatric complications like anxiety, feeding dysregulation, obsessive compulsive disorder. We have also established its role in the addiction, especially nicotine and ethanol addiction. And from last few years, not more than 5 years, we have started working on its feasibility or its delivery system related to neurodevelopmental disorders, especially autism, and neurodegenerative disorders like AD or HD. I'm very happy to say that based upon our literature, it has been translated into clinical application, and nowadays doctors are prescribing agmatin-based medications for the neurological complications associated with the stroke. Uh, Two important outcome of our research projects. One is in the alcohol addiction and Alzheimer disease, where agmatin has been directly projected as a therapeutic entity for the reversal of alcoholism as well as for the symptomatic relief in the Alzheimer disease. These are the projects which are funded by different funding agencies with a total outlay of 4.5 crores. You can see the list. Most of the projects funded by different funding agencies, only one thing remain constant, and that is agmatin. We are working on this molecule and it has given us everything. As I'm telling you this repeatedly, it has given us everything. So starting from its dysregulation to restoring it for the therapeutic benefits of psychiatric disorders, we are working on it, and we have still like halfway mark or contributed very little, and we are expecting much out of it in due course of a time. So this has been associated with the development of the sophisticated laboratories. We have everything which is required for the neuropharmacology research, neuroscience research, and considered as one of the best neuropharmacology laboratories of the region. Associated with operant conditioning chamber, motorized stereotaxic apparatus for the intracranial and region-specific drug administration. We have fluorescence microscope, we have treadmill, we have everything which is required for the neuroscience research apart from all the regular sophisticated equipments. Thanks to our collaborators, Dr. Dada Saheb Kokre from UDPS Nagpur, Dr. Vandana Patrawade from ICT Mumbai, Dr. Nandakishore Godgale from Government College of Pharmacy Amravati, and Dr. Sushruta Kopula from Korea for their continuous support in this research journey. I'm proud to say this is all because of this excellent team of the investigator and co-investigator. We are working under the blessings of Dr. C.T. Chopde sir. He is a torchbearer of neuroscience research. We are just following his legacy. Well assisted, supported by Dr. Manish Aglave, Dr. Shubhda Mangrulkar, Dr. Madhura Dikshit, Dr. Mayur Kale, Dr. Sandeep, and Dr. Nitu Ankhede. I must mention here the contribution of Dr. Aman Upgannawar and Dr. Manoj Mahajan for developing two of my gems, Dr. Mayur and Dr. Neetu, to the extent that themselves they are getting funded by ICMR or even DHR. Thank you very much, sir. Thank you everyone for your patient listening.
Manoj sir, you are muted.
Thank you very much, sir. Brijesh sir, for this insightful and thought-provoking as well as practical session for our audience. Now it's request to the participants, if they have any queries, sir is there. You can ask your queries. Okay. So thank you, sir, for your guidance on grant writing and funding opportunities, especially with ICMR, focusing on pathway for research grant proposal preparation and its submission. It has certainly provided clarity and direction to faculty members, researchers, as well as students aspiring to pursue the funded research. It's now our pleasure to present the online certificate of appreciation to Dr. Brijesh Taksande as a token of our gratitude for his valuable contribution to day 5 of this FDP SDP. Sir, we sincerely thank you for sharing your expertise and experience with us. Thank you, sir. And I'd like to request to be remain there for the valedictory function of this 5-day FDP. Now we are moving towards the concluding part of this enriching academic journey. We now begin with the valedictory function of this 5-day online FDP and SDP, titled Innovative Approaches and Emerging Trends in Pharmacology Teaching, Research and Practice. Over the last 5 days, this FDP has successfully brought together the academicians, researchers, students, and industry professionals on a common platform to deliberate on contemporary challenges and emerging trends in pharmacology. The enthusiastic participation and academic engagement from all of you truly reflect the success of this program. We are honored to have with us Dr. C.D. Upasni, Principal of SNJB Sriman Suresh Jain College of Pharmacy, presiding over this function, and Dr. Prakash Wadkar as the guest of honor for the valedictory session. We are also pleased to have with us Dr. Brijesh Taksande, Dr. Aman Upgannawar, who is head professor and head Department of Pharmacology and also the coordinator of this FDP. Before we proceed to the valedictory address, we would like to hear from participants and their experiences. The delegates, anybody is interested, kindly share your views on this 5-day FDP SDP program. Okay. Also, several delegates shared short videos with their messages highlighting their learning outcomes and experiences from this FDP. Let us know now take a few moments to watch delegate feedback videos.
Hello everyone. This is Dr. Trupti Deshpande, Professor and HOD of Pharmacology Department of JSPM's Rajarshi Shahu College of Pharmacy and Research, Tathode, Pune. A big thank you to Honorable Dr. Upasni sir and Dr. Aman sir for giving me this opportunity. Dalai Lama once said, share your knowledge because this is the only way to achieve immortality. And are really very fortunate. We have got this opportunity of 5 days FTP program organized by Sri Suresh Datta Jain College of Pharmacy, Chandwad. We are really very thankful, sir, for organizing these wonderful sessions for these budding pharmacists, pharmacologists particularly. The sessions were very relevant, the topics were very relevant, very well organized FTP it was. The speakers were truly enthusiastic, very experienced and knowledgeable one. They are very down to earth as well. We got to know, we got to learn many newer skills from them. Hands-on training also provided for many newer tools and techniques. What you can say, the interactive sessions and truly engaging sessions made this event truly a success. We got to know about Copilot, Gemini, newer advanced techniques of pedagogy by Pulshettiwa sir. Archana ma'am let us know about the newer techniques in phytopharmacology. Bhavin Vyas sir told us about advances in biostatistics. We also got to know about network pharmacology, molecular docking by Patel sir. So all these techniques are really high time need of this era, like AI era, or you can say NEP era. And we are really thankful for the same. Uh, Thank you so much for organizing such wonderful FTP. Looking forward for more events and also would like to contribute from our side also. Thank you so much, sir.
Hello everyone. Greetings of the day to one and all present over here. I am Dr. Shubhangi Pawar from Department of Pharmacology, MGB's Pharmacy College, Panchavati, Nasik. I am happy to share my review about this FTP. This 5 days FTP enabled me to integrate new approaches, new computational techniques in my current academic understanding. For current learners, pedagogy is merely not teaching from the book or not delivering content from the book, but integration of AI tools, different pedagogical technique is essential for the current learners to effectively deliver our content. Similarly, in case of research, this FTP emphasized that scientific validation through the AI screening techniques or network pharmacological approach had become so essential to strengthen the credibility of our research and to increase the acceptance of our research. I would like to congratulate respected convener and the coordinators and entire team of SNJP's SSDJ College of Pharmacy for the successful conduct of this FTP. In future, if I could work on network pharmacology or molecular talking type of concept, definitely the credit will go to this FTP, which has sensitized me to do so. So thank you so much for this FTP.
Hello everyone, I am Mayuri Injagtap. I am working as an assistant professor at a given like Institute of Pharmaceutical Education and Research, Nasik. Also, I am a research scholar from Mate Institute of Pharmacy. I am an alumni of SNJP, and being a part of alumni, it was my great pleasure to be part of this FTP and SDP of the SNJP College of Pharmacy. I would like to say thank you to all the coordinators, all the organizing people, and all the resource persons who have given their all efforts for this program, and they have given best of best to us. Because of that, the session went good, and we came to know how we can use various tools for our learning purpose also and for our research purpose also. It was not just for us, it was as a faculty, it was a great that I can use those tools to teach my students how they can learn new concepts. And these are efforts I am seeing of SNJB since I was a student, and now it's been more than 4 years. I am a part of SNJP, and every time they are having something new to give to their students and all the peoples from pharmacy field and other than pharmacy, how they can explore themselves in the research and how they can contribute to the world by learning a new things and adding some beautiful things in the research. And I'm really thankful to everyone. Thank you so much.
Hello everyone, myself Ankita Gupta.
I study at Mandish Institute of Pharmaceutical Science and Research Center of Moscow. And now recently I am working as a lecturer in Neurologic Shikshan Samstha, College of Pharmacy, Meda. My experience about FDP and SDP is are essential for maintaining a high academic standard. They are most effective and they are practical, continuous, and aligned with institutional needs, as well as SDP is crucial role in preparing students for real world challenges, especially when they are interacting with world and student cellular. Everyone.
Hello, good evening everyone. I am Mr. Varun Vishwet Joshi, Assistant Professor from the Department of Pharmacology, Dr. Golpe Institute of Pharmacy, Kolpewat, and a research scholar at Research Center, Department of Pharmacology, SNJB, Srimal Suryadada Jain College of Pharmacy, Chandra. These 5 days FDP on innovative approaches and emerging trends in the pharmacology, teaching, research, and practices was highly informative and well organized. The sessions provided the practical insights into the modern teaching and research trends. I sincerely appreciate the efforts of the organizing team and would be interested in the participating in the similar activities in the future. Thank you. Namaskar, myself Rishikesh Willis. I am working as assistant professor and head of department pharmacology at Amulbani College of Pharmacy. As a participant of the 5 days faculty development program, it's my pleasure to give my view on this program. So this program was organized by SNGB's College of Pharmacy, Sarwar, and it was a very unique opportunity for all the fellow pharmacologists to take advantage of this faculty development program. During these 5 days, we have learned many new things like AI tools introduced by Dr. Pulshi Shettiwar was very good opportunity to learn new tools. Then Dr. Bhavin Vyas, he gave valuable insights into statistical methods like R programming. Then Dr. Archana Negi, she gave the latest insight into the phytopharmaceuticals applied in drug discovery. Then Mr. Siddharth Patel also introduced a new field for us like molecular docking and its application in new drug discovery. Then Dr. Tarsade also gave very valuable information related to research paper writing, grant writing. So this type of faculty development program is need of hour for all these current pharmacologists as well as future pharmacologists. So I congratulate the complete team of SNGP's College of Pharmacy, Chandwad, Principal Dr. C.D. Upasi sir, Coordinator Dr. Aman sir, and Manoj Mahajan sir. I congratulate all the team members for organizing this type of faculty development programs. In future also this type of programs are needed for development of the faculty. Thank you.
Thank you all the participants who shared their thoughtful reflections. Your feedback is extremely valuable and motivating. Now I'd like to request Dr. Aman Upanglawa, coordinator of the FDP SDP, to kindly share a brief reflection on the program and also introduce our guest of honor for the valedictory session. Over to you, Aman sir.
Okay, thank you, Dr. Manoj. And dear participants, resource person, and faculty members, my dear colleagues. Today marks the concluding day of our 5 days online faculty development program and student development program. Over the past week, we have witnessed an enriching journey filled with brainstorming sessions, insightful lectures, and enthusiastic participation from all attendees. Few of the highlights of the programs are: the day one started with commence with a keynote address by our honorable principal, Dr. C.D. Upasni, who emphasized the changing landscape of pharmacology education in the competency-based era. This was followed by a talk with Dr. Satish Polshettiwar, Professor and Head Department of Pharmaceutics, MIT World Peace University, Pune, who delivered an engaging session on digital pedagogical tools in pharmacology, covering applications of virtual labs, gamification, and AR/VR concept. We were privileged to hear Dr. Bhavin Vyas on day 2, professor at Maliba College of Pharmacy, Bardoli. He provided a detailed discussion on data analysis and biostatistics in pharmacological research with practical demonstration using GraphPad and R software. Day 3 session was graced by Dr. Archana Negi, Professor and former Dean of Humayun University, Nainital. She spoke on phytopharmacology and natural products as a future therapeutics, elaborating on the fascinating journey of herbs from traditional medicine to modern application. Day 4 shared by Dr. Mr. Siddharth Patel, Assistant Professor at NMIMS Mumbai, shared his expertise on network pharmacology, docking and molecular dynamics, MD stimulation, offering practical insights into this advanced technology and techniques. Day 5 today we had the honor of listening to Dr. Brijesh Taksande, Professor and Head at Kishoritai Boyer College of Pharmacy, Kamti. His talk on grant writing and funding opportunities was highly valuable, providing guidance on securing research supports and advancing academic projects. As we move towards the valedictory function, we are delighted to welcome our guest of honor, Dr. Prakash Wadkar sir. It is my privilege to introduce Dr. Prakash Wadkar sir, who presents adds great value to this occasion. We extend our heartfelt gratitude to him for joining us today and for his continued support in fostering academic excellence. It is my privilege to introduce Dr. Prakash Wadkar, a distinguished leader in the field of toxicology and pharmacology. With over 28 years of experience in preclinical safety evaluation and laboratory animal breeding facility, Dr. Prakash Wadkar is currently serves as Senior General Manager and Deputy Test Facility Manager at Bharat Serum and Vaccines Limited, Mumbai, a part of the Mankind Group. Since joining Bharat Serum and Vaccine in 2007, he has played a pivotal role in shaping world-class animal testing laboratories and ensuring compliance with international standards. His career spans STEM institutes including the Indian Institute of Toxicology, IIT Pune, and the Institute of Toxicological Studies, Pune, where he contributed significantly to advancing regulatory toxicology and pharmacology practices. Among his many achievements, Dr. Prakash Wadkar has successfully achieved AAA LAC accreditation and GLP certification from BSV Animal Testing Laboratory. He has established a state-of-art laboratory aligned with global standard in 2021. He has contributed to regulatory submissions across agencies such as USFDA, EMA, ANVISA, and WHO. Been honored with the Outstanding Pharmaceutical Scientist Award in 2021 for his remarkable contributions to toxicology and pharmacology. He has delivered numerous lectures and sessions on preclinical safety evaluation at academic and industrial platforms. He is widely recognized for his expertise in regulatory toxicology, GLP, ALAC, compliance, preclinical safety, laboratory animal science, and leadership and mentorship. His vision and dedication have not only advanced scientific standards but also inspired and guided the next generation of researchers. We are truly honored to have him with us. Please join me in extending a warm welcome to Dr. Prakash Wadkar. Now, may I take this opportunity to respectfully invite Dr. Prakash Wadkar to kindly share his valuable insight with us. Sir, we would be honored if you could guide us from your rich experience on the industrial perspective, regulatory aspects, and career opportunities in pharmacological research. Your thoughts will surely inspire and enlighten our participants as they look forward to shaping their professional journey. I welcome you, sir, and over to you.
Thank you, Aman sir. Good afternoon, good evening, esteemed organizers. Am I audible?
Yes, sir.
Good evening, all, and good evening, esteemed organizers, distinguished resource persons, honorable faculty members, dear students, and all participants. First, I would like to extend my sincere gratitude to respected Professor and Principal Dr. Chandrasekhar Upasni sir and the entire institutional leadership for providing this valuable platform to collaborate and explore the potential of their team. I would also like to express my gratitude to an influential personality, professor and head of Department of Pharmacology, Dr. Aman sir, and the organizing committee for their dedicated efforts in planning and executing this innovative program. It gives me immense pleasure and privilege to deliver the valedictory address for this faculty student development program titled Innovative Approaches and Emerging Trends in Pharmacology, Teaching, Research, and Practice. Pharmacology as a discipline continues to evolve in response to scientific advancements, healthcare needs, and societal expectations. I believe a key strength of this program has been its faculty student collaborative approach, which fostered a shared learning experiment. Faculty members gain fresh perspective from the curiosity of students, while students benefited immensely from the experience, mentorship, and academic insights of their teachers. The mutual exchange truly reflects the spirit of academic development. I'll briefly speak on industry perspectives, regulatory aspects, and career opportunities in pharmacological research. So, in industry, the challenge pharmaceutical research is a backbone of modern healthcare. So, there are emerging trends we are witnessing of that emerging trends. For example, AI and data driven discovery. Earlier, people used to do a manual processing and it cost more, a time consuming method. So now the trends are there in the industry. AI is accelerating molecule screening, identify potential drug candidates quickly, reduce its costs and time, personalized treatment based on patient data. Then there is a precision pharmacology, which personalized medicine is no longer just a concept, it's a reality. So it helps avoid side effects, improve treatment effectiveness, and personalized therapy for each patient. Then there is a digital manufacturing. In digital manufacturing, there are 3D concepts. So 3D concepts customize those forms to individual persons, like there are tablets, there are pills, which we can give exact doses to the particular patients. Then there are traceability in supply chain. While manufacturing practices, after manufacturing, the drug must be entered into the market. When it enters into the market, supply chain plays an important role. So supply chain starts from production to the drug enters into the market. It is fastened process nowadays because of the traceability. Then we will see the emerging concepts like biologics and biosimilars are there. So moving beyond traditional small molecules, now complex biological therapies are there, which complex biological molecules are like proteins, antibodies, and vaccines. So these are the drugs which we can utilize for cure various complicated diseases like cancer, autoimmune disorders, and diabetes. So I'll give you some examples. We are working on, since BSV is a leading biopharmaceutical company, so we have gone through a lot of experiments and lot of products we have prepared, and these products there are in the market and leading life-saving drugs, you can say directly. So some drugs are there under biologics. For example, recombinant anti-D. So recombinant anti-D earlier, the products which made from the human plasma or human oriented product, you can say the human plasma required to preparation of this kind of drug. But now the trend has been changed, and the trend we can see we have utilized the recombinant technology. So CHO cells we have utilized, and under CHO cells we can produce maximum amount of production, as well as the diseases like HIV virus, you can say, or HBV virus, or all other critical virus screenings. We can avoid that virus screening, which carries from human plasma to the products. So here CHO technology, like Chinese hamster ovary technology emerged, and with the help of this technology, we can produce a number of recombinant technology products. So Arentine is one of the product which used as a life-saving product for Rh positive. You must have aware about this product. So the mother having Rh positive factor, like blood groups having a positive and negative factors. If suppose mother having positive factor, a positive negative blood group, and the fetus having a positive blood group, in that case, the maternal blood, the fetus blood may be mixed into maternal blood, and in that case, the complications happen. To avoid that complications, we need to take this kind of therapies. So you can say this kind of therapies are life-saving products. Then one more product I can put in front of you, like ASVS. You must have aware about anti-snake venom product. So anti-snake venom is one of our product, and we have seen series of development in this particular product. So while developing this kind of products, we have seen a lot of changes. Earlier, the monovalent products were there. So the monovalent products, when we give to the patients at the time, see if you see in India, there are 4 venomous snakes are present, like krait, cobra, Brussels viper, and soft-scale viper. So in that case, when we give the ASVS product, earlier we used to use a monovalent product, so specific product for specific snakes. But in a current trends, if you see, the polyvalent products are available in the market, irrespective of whichever snakes it may be. Like krait and cobra is neurotoxic, and Brussels viper and soft-scale viper are hemotoxic. So irrespective of whichever snake it may be, you can give a polyvalent therapy. So polyvalent products are available in the market in a current scenario. So these are all development happens over a period of time. Apart from biologics, there are other products also in the market. For example, liposomal amphotericin B. You must have aware about this product in COVID-19. Post-COVID complications, the mucormycosis happened. So for mucormycosis, we used to use this type of product for post-COVID complications on mucormycosis. So liposomal amphotericin, earlier it came into powder form, but that powder form, when it enters into the body, it creates severe problems to the patients, like maximum, you can say maximum side effects. So maximum side effects means it's a RBC toxic drug. When it enters into the body, it lyses RBCs, and it creates severe problems to the RBCs. So later on, it developed into the form of emulsion, and after emulsion, in a current scenario, if you see the liposomal amphotericin, you can find in a liposomal forms, it creates a very low toxicity, and its efficacy is comparatively high. Then one more product I can say, Luprodex. Luprodex is one of the product which earlier we used for a prostate cancer. 1 month depot injection was there. So when you take this particular injection, it will sustain up to 1 month. But later on, the enhancement happen in the drug, the development happens in the drug. Then 3-month depot injections developed. Then later on, in current scenario, if you see, the 6 months depot injections also available in the market. So you can utilize the 6 months depot injections for 6 month duration, and slowly it will release in the body of patients. So this kind of development happens in a gradual manner. So some biosimilars are there. So biosimilars also emerging trend in the market now. So biosimilars means you can say nowadays we are working on some generic products. So whenever the patents expires, you can work on different biosimilars. It will helps to release products in the market in a shorter span and in a less, you can say, less expensive. So we have to show the biosimilarity and we have to show its efficacy, and you can release this kind of drug in a 1 to 3 years of span in the market. So uh, this can be also an emerging trend in the market. So biosimilars, we can see the biosimilar is emerging trend. Sustainability and environmental responsibility. In pharma, the EHS concepts evolved uh, in a current scenario, environmental health safety. So environmental health safety shows it's like uh, sustainability, you can say directly, for the safe planet and future generations. So if you see the previous scenario, people used to like people used to use different kind of raw materials for preparation of drug, and lot of pollutions, uh, mm, uh, lot of pollutions and lot of like things were happening. So to reduce this kind of things, the greener concept or greener practices adopted by the industry, and uh, you can say eco-friendly chemicals people are using, reducing water, reducing energy consumption, and recycling materials nowadays people are using. So some challenges are there in the industry. See, whenever you are running in the running R&Ds in a large pharmaceutical company, pharmaceutical company, so we have to spend a lot of money to to maintain their R&D. So high R&D costs are there to develop the product. If you see, India is a country which majorly producing a generic products, but nowadays the concept has been changed, and India is like producing lot. India is like thinking to develop a novel products also. So a lot of companies are working to produce a novel products. But whenever you are going to produce novel products, the novel products require 10 to 15 years to complete their phases, different phases from the identification of molecules to enter into the market. And the another important factor here is uh, ratio of the success is very less. But the scenario has been changed in the current days. Earlier, before 2022, if you see, the scenario was different. The 5% to 6% rate was there. But in current scenario, the rate has been increased, and rate, if you see the novel product enters into the market 10 to 15% rate uh, in a current time. Again, here the challenge, long timelines, then compliances also. To maintain compliances, nowadays the compliances are very stringent. So to maintain that compliances, uh, these are the challenges, you can say directly, while developing a new product and entry into the market. But yes, there are opportunities. The institute like you, the pharmacy institutes or all other veterinary colleges and all those things. Here there is a gap in India. If you see I have seen there is a gap. Like mm, if you see the international authorities or international regulatories, they accept the data under GLP condition, under GCP conditions, or all other guidelines you need to follow. And for that purpose, you have to have a state of art facilities. So facility expectations and all equipments and instruments expectations are very high from the industry. But in a institution level, you can say institution level, that much awareness is not there, and because of that the collaboration is difficult with the private industry to educational institutes. So, but yes, opportunities are there. For example, the industry is gradually connecting with academia and research institute to share the knowledge, infrastructure, and talent in India. In India, there is a huge advantage. You can say it's a global hub because of the because of the scientific base and cost advantage. You can say there are CROs which are emerging CROs. You can say and established CROs also there. When we when I entered into the industry in 1997, at that time there were 4 or 5 different CROs were there, and out of these 4 or 5 CROs, 3 CROs were a GLP CRO. But in current scenario, if you see more than 150 CROs are there in India, and out of 150 CROs, near about 65 CROs are under GLP. You can say GLP accredited laboratories are there. So yes, opportunities are there, but we need to develop our infrastructure in such a way so that there should be all essential equipments, all essential instruments, and the state-of-art facility should be there. Then industry will come and collaborate with the academics institutions or some other institutions. And when it happens, then the India will reach at certain level to develop a maximum amount of, you can say, product, and we can supply a large amount of products all over globe. Then we will move to the regulatory aspects. So regulatory aspects, we can say regulatory agencies are the gatekeepers of innovation. So we are talking about innovations, but when we see innovative approaches are there, but whenever you are developing your products, there are regulatory agencies, and we need to cross that regulatory agencies to get it approve our products in the regulatory bodies. So regulatory framework safeguards innovation and ensure patient safety. Innovation must align with compliance. So compliance should be there. If non-compliances are there, then it would be difficult to get it approved our product under regulatory. So if you see the global scenario, global regulatory frameworks, the rules and guidelines that make sure medicines are safe, effective, as well as high quality products should be there before it reach into the patients. So different agencies are there all over world. For example, US FDA. US FDA, you must have aware about that. US FDA is a very stringent regulatory. When you crack the US FDA, you can send your drug, or you can enter into any country of the world. Then EMA is there. EMA, which related to Europe. So EMA, we can say a maximum European countries which comes under EMA. Then INVISA is there. INVISA is related to Brazil. INVIMA is related to Colombia. Then PICS are there, which related to Ukraine authority. TG is Australian authority. DGMED is the Peru authority. NPR is Malaysian authority. Then RCGM is an Indian authority which regulates the recombinant products, like whatever recombinant DNA technology products you are producing, you need to get it approved, your study plans from the RCGM, and again you have to provide your data, like whatever reports you are generating at your facility, you have to provide it to, you have to submit, or you have to present it to RCGM and get it approved under RCGM, and then only you will get approval for clinical trials. Then CDSCO is the body. CDSCO, which controls all other drugs apart from recombinant products, ensures safety and efficacy through rigorous guidelines, like ICH is there, which is international guideline. Then OECD is there, which comes with the GLP. So GLP is one of the important regulatory which includes 45 different countries. Under 45 different countries, India is one of the adherent country attached to the OECD GLP countries, you can say. Then the ISO guidelines are there for the medical devices. NTCT guideline is there. which whatever products you have to submit it to Indian regulatory, we have to follow NDCT guidelines, new drug and clinical trials. GLP is the accreditation which you have to apply in a preclinical research. Then GCP is there for clinical trials. Then the pharmacovigilance means post-marketing surveillance. So post-marketing surveillance, pharmacovigilance is there, and pharmacovigilance will take care of that all activities. Once the product enters into the market, what happens with this product, how it effective, the side effects and all those things which we have to collect, and the pharmacovigilance is the area which takes care of all those things. Then there are emerging challenges. So whenever your products you are submitting it to regulatory, so regulators are the people, they are like us only. So they need to take trainings. Like nowadays AI driven tools are available, then gene therapies like products are available, then digital health products are there. So people are using various types of technologies, and these technologies people are using to produce their drugs, to like anywhere in the anywhere in your areas, like preclinical areas, you can say clinical areas, you can say, or all other areas, people are using different kinds of tools which are available in the market. And for that purpose, the understanding of these dynamics is very critical for any entering research or regulatory affairs. So whenever you are submitting your data to the regulatory bodies, there must be the educated regulators should be there. I'll give you one example. So we were there in stem cell research earlier at 2010 and 2016. One of our subsidiary company was there, and we were working on various types of stem cells, like mesenchymal stem cells. Some stromal vascular fractions were there. So we worked on that, and when we submitted into regulatory, at that time there were no people in the regulatory who understand the proper concept of that stem cells. So in that case, the chances of rejections or chances of asking maximum requirements may possible. So in that case, so these are the challenges they need to take care, and everyone needs to be like educated in terms of whoever there are in the industry, they are developing their products, as well as whoever they are in the industry, regulatory, they should be aware about that tools and all those things. Then role of regulatory affairs professionals. So regulatory affairs professionals, what they do, they are the bridge between science and law. Means whatever you are doing as a scientist at your company, you need to submit your data to the regulatory affairs professionals, and that regulatory affairs professionals will see that data, will collect that data, and the data needs to be captured in the form of various forms are there, depends upon where you are going to submit it into. For example, IBs are there, investigational brochures, then common technical documents, CTDs are there, then dossiers are there. So we have to prepare these kind of documents, and we need to submit it to different regulatories to get it approved. So regulatory professionals or regulatory affair people, they are the bridge in between the scientists who are working on the products, and they collaborate. You can say, or they ensures our product will be approved, and it will be in the marketable. It will be in the marketable place in a stipulated time frame. Then emerging focus like pharmacovigilance is there. Pharmacovigilance which monitors medicines after they are approved. Then the important things we need to mention, like earlier, the GLP concept came into existence because of data integrity. If you see all over world, it initially the incidents happened in the US and Europe. So they started developing the stringent stringent, you can see, accreditation like GLP. So under GLP, you can maintain a data integrity, like whatever data you are generating, it should be accurate, complete, and trustworthy. Then the ethical standard. So ethical standard, your data should be fair and honest. So your practices should be fair and honest. For that purpose, if you see, whenever you are going to develop your facility, you need to have a CCSCA accreditation. So CCSCA, what CCSCA usually does, CCSCA gives approval to your facility, and you have to maintain Form A, Form B, Form C, Form D, and Form E. You people are, you must have aware about that, because everyone who are having this kind of facility needs to maintain this kind of forms and this kind of formats and everything. So the roles of regulatory affairs. RA professional ensures compliance across life cycle stage. Means whenever you are working on your drug, the drug should be provided to IND, CTA, means IND applications, CTA clinical trial applications means investigation drug applications, CTA means clinical trial applications, to the marketing authorizations. So then the career opportunities. So we'll move to career opportunities where we fit in. So we'll go to a career option. So research and development is there, in which you can enter into drug discovery scientist, formulation experts, preclinical pharmacology and toxicology testing, clinical pharmacologists, then clinical trials and pharmacovigilance, the roles like designing of clinical trials, monitoring and safety, regulatory affairs, quality assurance, RA specialist, you can enter into RA specialist, quality auditors, strategic RA advisors. Then digital and data sciences, AI modeling, you can enter into AI modeling, bioinformatics, digital biomarkers. Then you can enter into manufacturing, product development, and market functions also, like quality control. Then we will move to the closing of this program. So as we look ahead, remember that innovation is a continuous process, not a destination. The trends we explore today will keep evolving, and it's our responsibility to apply these insights in classroom, laboratories. Let's nurture a culture where teaching inspires research and research informs practice. Innovation in pharmacology isn't just about discovering new drugs, it's about creating solutions that improve lives globally. Each of us has a role in this mission. Let's turn innovation into impact. The future of pharmacology will shape not only by technology, but by minds that dare to innovate and hearts committed to making a difference. Together, let's be those minds and hearts. As we conclude, carry forward these insights and implement innovative approaches in teaching, research, and practice. May this program be a foundation of continuous learning, collaboration, and excellence. Stay curious, ethical, and committed to excellence. The future of healthcare depends on innovators like you, and I look forward to seeing many of you lead the next wave of breakthroughs. Thank you, and I wish you all a successful and fulfilling career. Thank you.
Thank you very much, sir, for your enlightening and motivating valedictory address. Your industry perspective and experiences have added great value to this FDP SDP and inspired all participants. Thank you once again. I now respectfully invite convener of this program, our beloved principal and president of this session, Dr. Chandrasekhar Upasni sir, to kindly address the gathering and offer his presidential address.
Thank you very much, Manoj, and good evening to all. Good evening, Dr. Prakash Wadkar, sir, for present here on this small FDP program. We at SNJB Shriman Surya Dada Jain College of Pharmacy have organized this 5-day online faculty and student development program with the theme of innovative approaches and emerging trends in pharmacology teaching, research, and practice. So this FDP was organized very well by the coordinators, Dr. Aman and Dr. Manoj, touching all the heads or objectives of this HDP. We talked about teaching, we talked about research, and we talked about practice also. And some new approaches have been explained by the resource persons. Very well said, Dr. Prakash Wadkar, sir, about the current scenario of the industry and pharmacology. We all were excited initially for this online FDP, and we were skeptical also how many people, with what sincerity they will attend and all those things, but we could get very good response. See, this is a challenge. We are entering into the National Education Policy 2020, which is called as new NEP also. And newer technologies, newer methodologies, newer pedagogies has to be introduced to make our subject interesting, and the students should be attracted to our subject so that many pharmacologists, sincere pharmacologists can be produced in this country, because need is too high. We have a very huge need for good pharmacology teacher and good pharmacology scientist also. Just teaching pharmacology in a routine way will not attract the students. We need to make the things interesting. We need to make the things very much fascinating for the students to grab their what you can say, vision, or to grab their mentality, or to grab their motivation towards the subject. And an interesting teacher or a versatile teacher can make the subject very interesting. So with this view and with this idea, we at SNJB, we are going into the autonomy in this current year, 26-27. We will be an autonomous institute. And as a part of the training of faculty members, we have organized this. What new we can do in our subjects. So fortunately, myself, Aman, Manoj, Rakesh, we are all from pharmacology. So they could initiate that process, and slowly this process will be initiated for all the subjects. So as a head of the institution, I must pay my high regards to the resource persons, those who came here, especially Brijesh and Dr. Brijesh and Dr. Prakashji, who are present here today. And thank you all faculty members for attending this FDP for 5 days continuously. Thank you very much. Over to you, Manoj.
Manoj, you are muted.
Thank you, sir, for your visionary leadership, constant support, constant encouragement, and whatever you are giving to us. That that has really fruitful for all of us. As we come to the conclusion of this valedictory session, it is my proud privilege to propose the vote of thanks. First and foremost, I express my sincere gratitude to our honorable management and trustees of SNJB's Sriman Surezada Jain College of Pharmacy, our leaders whose constant support, encouragement, and vision provides the foundation for organizing such meaningful academic initiatives. I extend my heartfelt thanks to our guest of honor, Dr. Prakash Watkar, for graciously accepting our invitation and for sharing his invaluable industry insight that have greatly enriched this FDP. I also express my deep sense of gratitude to our honorable principal, Dr. C.D. Upasni, for his constant guidance, encouragement, and unwavering support, which have been instrumental in successfully conducting this program. I sincerely thank all the distinguished resource persons, Dr. Pulshettiwar, Dr. Vyas, Dr. Negi, Professor Patel, and Dr. Taksande for generously sharing their knowledge and expertise in various domains of pharmacology. A special word of thanks to all the participants, faculty members, research scholars, and students for enthusiastic participation, academic discipline, and active engagement throughout these 5 days. Finally, I acknowledge SNJB Sriman Suresh Dada Jain College of Pharmacy for providing the academic platform and institutional support to conduct this FDP SDP. To the participants, the certificates will be issued uh, within the span of 5 days. With this, we formally conclude the 5-day online FDP SDP and looking forward to your continued association with us in future academic endeavors. Thank you. Thank you once again.
Thank you, Prakash ji. Thank you, Manoj.
Thank you, sir.
Thank you.
Thank you. Thank you.